All right. So there's what we're going to learn today. We're going to learn all about the anatomy of a skeletal muscle. This is all repeat from yesterday. So we learned all those terms yesterday, acute, chronic. All right, here's the big things of the day. So there are three types of muscles in the body. You have your cardiac muscle, is a little bit different. Again, this should all be review from your physio class. You have your smooth muscles, which will be a lot of your other internal organs. And then there's skeletal muscles. We will focus mostly on skeletal muscles today. All right, so here is kind of what a skeletal muscle looks like. It's hard to, it's not as good a picture as I would prefer that to be. So these would be microscopes. So cardiac muscles have some these nucleuses in the middle there. Smooth muscles are have again more nuclei in there. Your skeletal muscles actually are serrated almost, and that is how they actually contract and you move your body going as anytime you want to go somewhere. And we'll go into all the details on this today, and that's why this is kind of a tough one. So the anatomy. First, there is the entire muscle. So there's the surrounded by the epimysium. Anybody take midterm yet? If you have a chance to take medical terminology, it makes these classes a lot easier because epi means around, mysium is muscle. So if you get a quiz question up here, knowing those kind of things help a lot. So epimysium, around the muscle, and it's made of many bundles called fascioli. I can't pronounce things sometimes, so I apologize if I butcher how something is pronounced. All right, and then the next one will be individual muscle fibers. So I'll say that the fascioli are surrounded by the perimysium, so around the muscle. And then those are made up individual muscle fibers. Those individual muscle fibers are on the endo inside the mysium, and those are made up of myofibrils divided into sarcomeres. So we're just going bigger to smaller to smaller to smaller. We're kind of working our way down here. I'm happy there's lots of people playing basketball. All right. All right, so then there's the plasminelma, or meloma. So that would be the cell membranes with the outside of each individual cell, and those fuse each tendon together. They conduct the action potential, so that's the energy. This also maintains the pH in each of your muscles there and helps transport the nutrients. We'll go into tons of those details in Chapter 2. Then you're going to have your satellite cells. So those are involved in making your muscles grow bigger. So when you're trying to get stronger and get hypertrophy and grow your muscles by exercising, those are what are going to help with that. This is going to aid in response to injury, immobilization, and training. All right, getting smaller and smaller again. So the sarcoplasm serves as the cytoplasm of each muscle cell. And that's where all your glycogen is stored in the myoglobin. Those are your energy sources, which again, we'll do that in, I think, Chapter 5. We'll go over all the different energy sources for the body there. So just know that that's coming. Your transverse tubules serve as extensions to that plasminelma. And those carry the action potential deep into that muscle fiber. And I got a picture for all this in just a second. And then the sarcoplasmic reticulum is your calcium storage. So those are just all the terms that we're going to need to know. And there's still some more coming. But we'll go into more details about each individual one. So that's your vocabulary for the chapter. All right, so here's the picture of everything we just talked about. So you have the outer part of it, so each nucleus, and then it gets smaller down in. So we talked about the sarcoplasmic reticulum that transports things. Then there's the opening to the T-tubule in between each section. Each myofibril has all the different tendons together, like individuals. Sarcoplasm's around, the plasmanella contains everything. Mitochondria are your energy sources there, and then the nucleus for each. So this is all like one big cell section here. And then we're going to get even smaller into these blue and red markers here in just a second. So we talked about the sarcoplasm, transverse tubule, sarcoplasmic reticulum, and the plasmanella. And now we're going to zoom in on individual cells here. So myofibrils and sarcomeres. So going big, you have your full muscle, fascioli, muscle fiber, and then each individual myofibril. So again, we'll have this zoomed in. I'll have a bigger version of this in a little bit. So hundreds of thousands per muscle, and they run the whole length of each muscle. The sarcomere is the basic contractile element of each skeletal muscle, and it runs end to end of the full myofibril. So there's sarcomere is one section here. So from there to there, and then there's another one and another one and another one, and it just keeps going and going and going until it gets to the end where it attaches to a tendon.
Wait, what are the arrows for?
The arrows, oh, that's just giving you the bigger to smaller to smaller to smaller. Oh, in this chapter, if it's in blue, there's probably a quiz question in the bank about it. I only did that for the first like 2 chapters, and then I bailed on it when I first started teaching this class. But there's just a little hint for you on these first couple chapters. If it's in blue, there's probably a quiz question about it. And if that really helps you, I could add them in on other chapters too. But just let me know if that. Other classes have not said that helped them, so up to you guys if you need me to go through there. So each sarcomere has a distinct striped appearance, or kind of a striation is what that would be called. So the darker stripes are called A bands. The lighter stripes are called I bands. The H zone is the middle of each A band, and the M line is the middle of the H zone. So the H zone here is kind of this blue, I don't know how well you can see that up there. And this blue marker here. The M line is the middle of the H zone. And then you have your A band, and then the middle of that is the H zone. And then the M line is the middle of the other ones. So it's kind of whichever color it is there. And they're usually blue and red, but when you look at them under a microscope, you're going to have a lighter and darker. So a common boundary for each structure is it's called the Z-disc. All right, so protein filaments. So you have these tiny little things here. So you have your muscle contractions, and so you have your actin and myosin. And I have a really funny video here in a little bit that'll make you never forget what actin and myosin are. So they call the actin is the thin filament, and then the myosin are the thicker filaments. These show up darker, lighter. Those I bands contain only the actin filaments. And the myosin have the A band and the H zones. Give you a couple seconds there. There's just kind of the zoom. I forgot this. I don't think there's sound on this video. So this is just kind of that zoom in. So the actin and myosin are these two different sections here, and they interact to cause the muscle contraction. That one's kind of the boring one. I got a better one coming. So the myosin and actin are two intertwined filaments in each cell of the muscle, and there are thousands of them the whole length of the muscle. So they have a thing called the globular head, and it protrudes 360 degrees from the thick filament axis. And it will interact with the filaments for contraction. And then the stabilizer, to make sure they don't actually connect immediately or all the time, is the titin. So that gets in the way when you're relaxed or when you're decontracting, is that gets in the way and allows your muscle to stretch out again. And we'll talk more about titin in just a second. The actin is composed of three different proteins. So you have actin, tropomyosin, and tropamine. So actin contains the myosin bonding site. So over here in these pictures here. Tropomyosin covers it when at rest, and then tropamine moves the tropomyosin out of the way, allowing the actin and myosin to connect and pull on each other to get closer and closer together, causing your muscle contraction. And those are all anchored at the Z disc of each section. So Z disc and the H disc has the other one of the myosins. And they're equally spaced out by titin. Right, keeping going. So titin, nebulin is the anchoring protein. My apologies, there's one more little thing there. All right, so titin, this is the third filament in there. So this acts like a spring. So this is the stiffness that increases muscle activation and force development. It extends from the Z disc to the M band. Calcium binds to the titin, and we'll learn tomorrow that there's a thing that releases an action potential which releases calcium into the muscle or into each individual fiber, and then tells the muscle it binds to the titin, which increases the amount of force. Sometimes it's called the winding filament therapy. And really the thing you need to know about titin is that it prevents overstretching. So that's the part that's going to tell your body up too far, and then it's going to activate the pain receptors, which is going to go up through your spine and tell your brain, ouch, and then you'll stop. All right, I think this is this is not the funny guy. That's after our standing break. So the sliding film...
Cross-linking occurs by a myosin head comprised of multiple proteins capable of conformational and positional changes. The myosin heads bind actin binding sites and undergo a conformational rearrangement in their proteins. The conformational rearrangement rotates the position of the head and pulls the actin along the myosin. In the next scene, we will look at the biochemical events related to actin-myosin binding.
Which, I don't have that one there. So there's kind of the big picture of it all. So you get very large, get it a little smaller, then we zoom in on each individual one. So each nebulum, and then the actin looks like the little kind of bubbles there. And those bubbles grab or move aside, and then each of these will grab each other and get smaller and smaller and smaller, or to make the muscle contract. All right, standing break. You can do whatever you want this time. Oh, I like that. It just stands it up. I like that. If I had had an iPad in college, I would have loved taking notes like that. I would have downloaded the PowerPoints and just written on. That would been how I would have studied. You're not allowed to leave. Now, normally I leave it open, but since they were bouncing a ball out there, if you really need to leave, it's not a big deal. keys are over there. All right, so we got to keep moving. We got to get through a lot of stuff here. And this guy's
Romeo and Juliet, Helen in Paris, Tristan and Isolde. These famous star-crossed lovers bring to mind insatiable longing, forbidden love, and tragic separation. And poets and emo rockers love them for it. But you know where else you can find a nice, hot romance? Your muscle cells. They've got their own famous coupling, a pretty pair of tiny protein strands called actin and myosin. Romeo and Juliet may have set a chain of tragic events into motion with their infatuation, but deep down in the cells of your muscles, the hot protein action between actin and myosin is actually literally causing all of your motions. All of them. And I don't just mean voluntary stuff like walking down the street or moving your mouth so you can talk or chew chips, because your muscles also support your weight and help fend off gravity. The amazing thing about your complicated self-healing, blood-guzzling muscle tissues is that they turn chemical potential energy into mechanical energy, or movement, simply by doing two things: contracting and relaxing. And that contracting and relaxing is exactly what's fueled by the constant coupling and separation of biology's greatest lovers. Somebody get these proteins a movie contract.
If you want to look this guy up later, this will actually, I would probably watch this before you take your quiz, but it's this Anatomy and Physiology Crash Course is the guy's YouTube site.
You will recall from our early lessons on tissues that you're kept alive and moving by three types of muscle tissue: smooth, cardiac, and skeletal. Your smooth muscle tissue is found in the walls of all your hollow visceral organs like your stomach, but it gets its very own muscle tissue type, cardiac muscle, which looks involuntarily in Captain America. And those types, the ones you can see and feel and flex, are your 640 skeletal muscles. They're striated like cardiac muscle tissue, but they're also mostly voluntary, meaning you have to think about using them and activate them with your somatic nervous system. Most of them attach to your skeleton and create movement by pulling bones in different directions as they contract. Each one of your different skeletal muscles, like your biceps brachii or vastus lateralis or gluteus maximus, is technically its own organ, made up mostly of muscle tissue, but also of connective tissue, blood vessels, and nerve fibers. And because your muscles are voracious energy hogs, each one is rigged up with its own personal nerve to stimulate contraction, and its own artery and vein to keep it well fed with all the blood and oxygen and nutrients it needs to operate. But to understand those operations, we first need to get a grip on the anatomy of a skeletal muscle, which involves fibers within fibers and lots of layers. Basically, a skeletal muscle is constructed like a really sturdy piece of rope. Thousands of tiny parallel threads called myofibrils squished together to form muscle fibers, which are your actual muscle cells, cells with mitochondria, multiple nuclei, and a cellular membrane called a sarcolemma. Those muscle fibers then form larger string-like bundles called fascicles, which combine to form the larger rope-like muscle organ like your biceps brachii. Overall, this bundles of bundles configuration makes muscle tissue fairly sturdy, but considering how much abuse your muscles take when you do something like pretty simple, like lift a big bag of dog food, it's no surprise they need a little help. That's why every muscle contains a few different kinds of supportive sheaths of connective tissue, the protective reinforcements to keep that bulging muscle from bursting. So that is the structure part of the story. But if you want to get into the nitty-gritty, the down and dirty of how you actually move, well, there are rules. Really just two main rules, and they have to do with proteins. And they're both true for a lot of the proteins we talk about, whether they're enzymes or ion channels or receptors or muscle proteins. And these rules are: 1, proteins like to change shape when stuff binds to them, and 2, changing shapes can allow proteins to bind or unbind with other stuff. So keep those rules in mind while we see how a muscle fiber contracts and relaxes. Now, remember those tiny myofibrils that bundle up to form your muscle fibers? They're divided lengthwise into segments called sarcomeres, which contain 2 even tinier strands of protein, 2 different kinds of myofilaments called actin and myosin. And it's their angsty story of star-crossed love that fuels every movement your body could possibly dream up. A sarcomere contains both thin filaments made up mostly of 2 light and twisty actin strands, and thick filaments composed of thicker, lumpy-looking myosin strands. Each sarcomere is separated by what's known as a Z-line at either end, which is just a border formed by alternating thin filaments in a kind of zigzag pattern. A muscle contracting is all about sarcomeres contracting, bringing those Z-lines closer together. All right, so now comes the romance. When your muscle cells are at rest, your actin and myosin strands don't touch, but they really really want to. Specifically, that club-headed myosin wants to get all up close and personal with the actin. When this happens, and it will eventually, it's called the sliding filament model of muscle contraction. But in the meantime, like in any good love story, the pair have some obstacles to overcome. Namely, actin is blocked by a couple of protein bodyguards called tropomyosin and troponin, which keep getting in the way. Luckily, these guards can be bought off with a little ATP and some calcium. I'd prefer cash and nachos, but whatever. Remember, ATP is kind of like molecular currency. It contains chemical energy, and your muscles are all about converting chemical energy to motion, so they're always hungry for more ATP. Your muscle cells have lots of nuclei, but some of them also have a lot of mitochondria, whose sole purpose in life is to crank out ATP. And muscle cells also have their own version of an endoplasmic reticulum, the cell's transport and storage system, but in this case it's specialized, so it gets a special name, the sarcoplasmic reticulum. Its walls are loaded with calcium pumps, which use ATP to save up a bunch of calcium ions. And it's also studded with calcium channels that are linked to voltage-sensitive proteins in the membrane of the muscle cell. Say I want to move my arm. My brain sends an action potential along the motor neuron until it synapses with a muscle cell in my arm. The receptors on that muscle cell are ligand-gated sodium channels. So when the motor neuron releases our old friend acetylcholine into the synapse, the channels open up and create a rush of sodium into the cell as a graded potential, which, if it's strong enough, causes nearby voltage-gated sodium channels to open. Now I want to take a second and point out here that we're still talking about an action potential, but not in a neuron. This is happening in a muscle cell, people. So that action potential zips along a muscle cell's membrane, the sarcolemma, which has lots of tubes that run deep inside the cell called T-tubules. When the action potential travels down one of those tubes, it eventually triggers the voltage-sensitive proteins that are linked to those calcium channels in the cell's sarcoplasmic reticulum. When those channels are thrown open, the calcium stored inside rushes into the rest of the cell, and finally myosin is like, yes, here we go! At this point, the myosin is totally stoked, because the bodyguards that have been frustrating it are in for a big, irresistible distraction. That's because the protein troponin just loves to bind with calcium, and remember, when stuff binds to proteins, the proteins change shape. So the calcium latches onto the troponin and causes it to pull the other bodyguard protein, the tropomyosin, away from the sites on the actin strands that the myosin really wants to get its paws on. And suddenly it's all, okay. But the only myosin heads that can bind to those newly exposed sites are the ones that are ready for action. That is, the ones that have already grabbed a molecule of ATP that's been floating around and broken it down into ADP and the leftover phosphate. When a myosin head does that, it moves into an extended position, kind of like a stretched spring, still holding on to the ADP and the phosphate, and still storing the energy that was released when they were broken apart. So after all that, with the myosin primed for action and the bodyguards out of the way, the myosin finally binds to actin, and it is beautiful. When they bind, the myosin releases all that stored energy, and in the excitement of it all, the myosin changes shape. It pulls on its precious actin strand, kind of like pulling a rope hand over fist. In the process, it shrinks the whole sarcomere and contracts the muscle.
So there's still a lot more of that, but we just don't have time to watch the whole thing. Very much, there's a lot of our, I don't know if you noticed all the vocab basically that we just went through. He did a much better job of actually showing exactly where and what that was. So highly recommend find that video and watch it again.
Romeo and...
All right, so each motor unit has a alpha neuron and all the fibers, and those move together to have your contractile force. And he kind of mentioned there about the neuromuscular junction. That's the synapse there of the motor neuron and the muscle fiber. That connection communicates with the neuron and the muscle. So here would be the neurons in your brain going all the way down to the muscle, and they would tell it to contract basically by releasing calcium and releasing things in there. So a motor unit is an alpha. Each alpha motor neuron is let's see, what does it say? All alpha neuron are all of the muscle fibers are innervates, as in they're not going to cause pain. All right, so very quickly, this is all the things that he just said there, but you may want to know this just for the order here. So action potential starts in the brain. That potential goes down the axon terminal and releases acetylcholine, and then the acetylcholine crosses over the synapse, binds with the receptors in the plasmanella, and then that travels down to the plasmanella into the T-tubules. We'll go into more details on this tomorrow. And that triggers the release of calcium into the from the sarcoplasmic reticulum, and then the calcium enables the actin and myosin to contract. So it all goes pretty quickly Again, we've kind of already mentioned this. And then here's, I don't know if there's any talking here.
Excitation is how the nervous system stimulates an action potential in the muscle fiber sarcolemma. In order for a skeletal muscle to function, it must contract, which means it gets shorter. And when a muscle gets shorter, the skeleton moves at the joint it crosses. This is First, a wave of action potential spreads from the motor end plate in all directions. This is similar to the ripples formed on a pond when a stone is thrown. When this wave of excitation reaches the transverse tubules, which are commonly known as T-tubules, it continues down them into the sarcoplasm of the muscle fiber. Next, the action potential stimulates the opening of voltage-gated ion channels in the T-tubules. These channels are physically linked to calcium channels in the terminal cisternae of the sarcoplasmic reticulum. These calcium channels open as well. And since calcium ions are higher in concentration in the sarcoplasmic reticulum than they are in the sarcoplasm, they diffuse out and into the cytosol.
And then that allows the actin and myosin to interact and go. So there's basically saying the same thing here of all those things moving across. So coming from the motor neuron, releasing here, releases into the sarcoplasm, which then allows the actin and myosin to contract. And there's a fancy version of the whole thing that I kind of like. So, all the way from the action potential, same basic concept here, just kind of being said again. So the role of calcium in a muscle fiber is the action potential arrives to the T-tubule, makes an electrical charge, releases calcium. Calcium binds to tropamine, that's to get it out of the way, and that allows the actin and myosin to then bind and contraction. So we're saying this is a bunch of ways here. All right, keeping going. All right, so sliding movement theory. Again, we kind of gone over this in the video there, but we're just going to touch on it here. So in a relaxed state, the actin and myosin are not binding, and the myofilaments may overlap just a little bit. In a contracted state, that pulls them together, and now they are over. Everything is shortened and everything like that. So after the power stroke ends, the myosin detaches from the actin site. The myosin rotates back to its original position and attaches usually to another site further down. So they just keep kind of grabbing, causing that to happen. So that process goes until the Z-dick reaches all the way to the end, or the action potential stops, and calcium gets pumped back into the sarcoplasmic reticulum, allowing the muscle to release. All right, 10 seconds, stand up, standing break. Because I got about 20 slides and 15 minutes. It can be done. All right, let's keep going. So for energy for a muscle to contract. So we will talk about ATP a whole lot more and how it's made and the chemistry behind it. But let's just say that everything you eat eventually gets turned into ATP, and that's what is necessary to keep your body going. And in this case, to keep muscles to contract. So as the binds happen in the myosin head, the thing called ATPase is on the myosin head. That ATP, which is adenosine triphosphate, gets converted into adenosine diphosphate, so 2-tri, and then 2, because it loses one of those phosphates, and also energy comes out of there. And that is only for muscle contraction and relaxation there. So when the action potential ends, electrical stimulation in the sarcoplasmic reticulum stops. Calcium gets pumped back, and then that now it is stored until the next action potential arises, and then it's going to require ATP to pump it back as well.
For the SR, this is for
Sarcoplasmic reticulum. So without calcium, the tropamine and myosin return to resting, and then they don't allow the actin and myosin to touch anymore, or to bind anymore, and then it allows things to go back to a muscle relaxation state. Oh, I think this was those two gals that were kind of funny. I don't know if I have that much time, though.
Are big. Of course, generally cells are microscopic, but these muscle cells can have lengths up to 30 centimeters long. Anyway, inside a muscle fiber are multiple myofibrils. I tried to remove a simple area called the thick filaments are held by.
Yeah, we're gonna keep going.
Are big.
They're saying we've kind of said that 3 times now. So, all right. So next thing we need to know is, did that just blink? Okay, lovely. That just means all the clocks in my house just got reset. Dang it. All right, so there's 3 different types. Well, there's technically 2 with some subtypes. So there's type 1 muscle fibers. So about half of the muscle fibers that you have are type 1 fibers. So the peak tension on those is about 110 milliseconds. So that's how long it goes from the furthest length to the tightest length. And that's actually called a slow twitch fiber. So that would be the slower movement, the consistent movements that you have. Type 2, on the other hand, have a peak tension of 50 milliseconds. We call those the fast twitch. And those when I took this class, there was only 2. So like since probably 2007, I think is probably when I started taking this, maybe 2008, because I graduated in 2010. When I took this class, there were only 2. There was type 2A and type 2C. And they've added this other one here, X, in there after. So like in the last 15 years, they've actually decided that there were more options, basically. But basically, the type 2Cs are the fastest twitching ones, and there's very few of those in the average person. Now, there are genetics that can make it better for some and worse for some. For example, like marathon runners, the Kenyan people can run faster and longer. They actually have more type 1 muscles in their body than the average person. That's just genetics. That's just an example. So some people can get a competitive advantage doing that. So this is what that would look like in a microscope. So the black ones are type 1s. Type 2s are very white. So the lightest ones, and then the type 2X are the kind of more gray. So I know that's probably not the greatest picture up there. Honestly, that one's better than in the projector downstairs. So the variable speed that they talked about there, it said like one was 110, one was 50 milliseconds. So the variable speed of the myosin is the ATPase. That's a chemistry term there. So the fast ones are the fast muscle cycling, and the slower ones are the slower contracting. I know. So they would actually get this with a muscle biopsy. So that will take about 10 to 100 gram piece of muscle, and the thing will probably be like 2 millimeters thick. Like, it's a huge needle. And I'll show you a picture of it in just a second here. And then that biopsy is frozen, sliced, and then looked at under a microscope. So that poor athlete, like they'll take like a Division I or an Olympic athlete, and they're like, we want to see what your muscle fibers look like compared to the average person. So they will do this muscle fiber test on them. And they also will do an electro thing there so that you can kind of see the different ones by different sizes as well. We're going to skip this one and keep going. You got it. Yeah, so we got 10 minutes here, and I got a few more slides. We're going to skip that one. So there's what that needle looks like. Look how thick that beast is. That looks like it would hurt. I mean, I get poked and prodded all the times with all the medical tests that I've had over the years, and whew, man, that thing looks scary to me. So this one said, this is off of a elite female runner on their calf muscle, it looks like. So then they would put it into the needle would be put that piece of tissue, they would then freeze it and then slice it up and then look at it. Like, isn't that intense, huh? So the difference with that type 1 to type 2 muscle fibers, so the sarcoplasmic reticulum, those type 2 fibers, have more highly developed sarcoplasmic reticulums, and you can, I mean, at least chemistry-wise, you can train your muscles, and that's what you run for, and you do different kind of sprints to trying to train your muscles, and part of that is training the chemistry in those muscles to work faster or slower. So you can slightly train it. So the calcium release is faster in a type 2 muscle fiber, and sometimes up to 3 to 5 times faster. The type 1s have are less than 300. The type 2s are larger neurons, things to tell them what to do. So greater than 300 per fiber. And there's all the details. Make sure you know these blue ones here. Like the preferred one is a type 1, and the oxidative capacity of a type 1 is very high compared to the other ones. So how much oxygen they use, which makes sense. The slower twitch ones are the ones that you're probably going to, when you're trying to run a marathon or run a long distance, you're going to use the slower twitch fibers because they fatigue slower and they use more oxygen to energize them. Whereas a fast twitch, like trying to jump and dunk a basketball, that's the one quick burst and it doesn't use oxygen to use that energy source. And we'll get into that in more details later, but the oxidative capacity is high as one of your quiz questions. So type 2X are the fastest, type 2 are the middle ones, and then type 1s are the slower one on how much power they have. So there's more in that type X or 2X ones compared to 1 and type 2A and type 2C. So all fibers reach max power at about 20% of peak force. Just going to keep going here. Nope. And I kind of already mentioned this one. Everybody has a different ratio of the different kinds of fibers. And also how you work out and how you train will also affect how exactly they work and which one's more dominant in each person. So type 1s are going to be more in an endurance athlete, and type 2 are going to be more in power athletes. So the easiest way to think of that would be like a marathon runner and a power lifter, or like an Olympic lifter. So most of you that are you ones that are playing basketball, soccer, you actually are probably 50-50 or as close as you can, because sometimes you need more power, sometimes you need more endurance, depending on what your sport needs. But unless you're doing long runs or major lifts or major bouts of speed, football tends to be more dominant by a type 2 because they sprint hard for 10 seconds and then they have like 30 seconds, 45 seconds before the next play. So like your high-level athletes, they will train in football to go faster and try to train those type 2 ones more often. And there's just a bunch of numbers there. I don't think you need to know them immediately. Just knowing which one's the fastest, which one's the slowest. That's what you need to know there. The peak force, they were saying, at about 20% is when they have, so the initial push or the initial pull of the muscle at that 20% contraction is when they have the most power. So also that's when you're going to get the highest jump. So like when you're trying to dunk a basketball, you want that your peak powers in that first 20%. All right, couple more here. So type 1 fibers, they're going to be the ones that use aerobic energy, so oxygen energy. So that oxygen is required for ATP production, and that recruits for the low-intensity aerobic exercises and just daily activities so that you don't get tired. For those of you in reffing class, having a lot of good type 1 things when you got to stand up for 6 hours straight, reffing games back to back to back to back, your legs get tired. And so you definitely feel like that activities of daily living, like just trying to walk to the car after the game, and you're just like, oh. And you're a sophomore, you're a junior? Yeah. There's a big difference in me. My freshman year, I laughed at all the seniors after practice. Like they were all like groaning, and they're like 21, 22, 23. Then when I got to 21, 22, 23, I was like, I get it now.
I never got like sore before until like a couple months ago.
Yep, and it gets worse. I'm sorry to break it to you. I used to be able to do a 2-hour basketball practice, sit down, drink a Gatorade, and then turn around and play pickup ball for 2 hours. That did not happen my senior year. It got worse and worse and worse. So your type 2s are going to be ones that don't use, so they fatigue very quickly. So they don't have really an aerobic endurance. They're more anaerobic. So they produce their energy, ATP, anaerobically. Type 2A have more force than type 1s, and they fatigue faster than type 1s. Then those are used for those short, intense distances. So less than a mile. Is it 1,600 meters a mile? It's still pretty short. That'd be 4 trips around that track out there. So I'd say that's a mile run. So type 2As are intense distances of less than a mile. And then the other ones for a 100-meter sprint would be so the fastest man alive. Anybody watch the 100-meter dash this year? That was like a photo finish. That was incredible. So I was telling you this, genetics have a lot of factors. You can change a little bit by training, like 10% in fiber type, but you're it's pretty much what you're born with, but you can make a little bit of change there. So, and the older you get, the more you lose the fast twitch ones. So that's why when you play me in intramurals, I feel so slow compared to what I used to feel like in college. And I also can't out-jump anyone anymore. It is so annoying. I miss being able to jump. So method for alternating force. So the less force production is going to use fewer. More force is going to use more units. And here's kind of the order that things happen. Your body will try to use the type 1 first. When that's not enough, then they'll add the more faster twitch and faster twitch. And same concept here. And then the size of them, the smallest ones are the type 1s. The largest ones are the type 2X. So that's why in that picture you saw really big black fibers in the picture. And they will always recruit in the same order every time. And we already kind of said that. Success is affected by other factors, how good of cardiovascular system. The person's motivation will actually affect how much their muscles work as well. Kind of makes sense. How you train and how big your muscles are. Okay, a couple more here. These should be review here. So a static would be an isometric thing there, and we'll talk about that a lot more. So like if I'm trying to lift something and it doesn't move, but my muscles are contracting, that's an isometric motion. So the joint ankle doesn't change. The myosin and cross bridges don't slide, so they're trying to and they just can't. Dynamic would be the whole movement for that. So concentric is when you're pulling them together. Eccentric is when it's loosening. And if you're trying to actually slow down, like if you're doing a bicep curl and you pull it up and you just let it drop, you're not going to feel that much the next day. But if you pull it up and try to slowly pull it down, or if you're using actually the cables or any of the actual machines down there and not free weights, and you're trying to eccentrically not let it, so now you're telling your muscles, oh, don't let it go, don't let it go, don't let it go, don't let it go. And you're telling those actin and myosins and everything and all that. So it's actually causing muscle confusion with all of those proteins in there. And then that's the one the next day you're going to be like, oh man, I'm sore. And it's because you were actually working harder on the eccentric side. All right, generation of force, kind of already talked about that. Length, tension, the longer it is, the more, or if it's too short or too stretched, you're not going to have much force. So when you get to here, you don't have a lot of power. All right, and that's all I got because I do need to get out of here because our next class in here wants the board. All right, so you do have 2 quizzes tonight, and then we will have another lecture tomorrow. And then Thursday, you guys will present your journals. So you may want to look up a journal or two either tonight or tomorrow, just so you're ready, because you need to, tomorrow night, you will have a quiz, and you'll need to turn in that journal the night before.
So, we can really just Google a journal? Like, I'm still kind of confused on the directions. You said it was Thursday?
Thursday is when you present them. So on the actual assignment for journals, yeah, we have this class on Thursday. We don't have it on Friday.
The lab was Thursday. Am I thinking the wrong class?
Well, technically, lab is the 4th day, and that's just how one of the registrars did it. It's a 4-day-a-week class for you guys, but on there.
Are just all the directions on there?
All the directions are on here.
Okay. To be fair, I haven't read the directions.
The best place to look is actually our library website.
Oh, for journals? Yeah.
Yeah, just look up on, because that's where you're going to have the access to the, yeah, you could use Google Scholar, but it's tough.
No, yeah, that's fine, that's fine. Oh, can we get the
Yeah, let's let them in. As Miss Newdeck is standing out there, and most of you guys are probably in this class too. There's a lot of people in this. I'm kind of surprised she's not doing it downstairs. I think you have 18 people in this class. No, Giselle, you can walk them in. You can walk them in this classroom. I know. I didn't know that. Caleb tried to leave for something in my standing breaks, and he's like, never mind, I'm okay. I don't know, it's kind of weird. All right.
Hello.
Ooh, how you doing? Oh my goodness, I figured you guys would make it so hot in here.
It's super hot in here.
Oh my goodness.
Do you need a screen? No. Okay. So I don't think you use the screen very often, but I know once in a while.
When we get a rain out, we do. I don't think it's any better to open this door. It's hot out there, but maybe circulation. Ah,
No, it ain't any better out there. It's worse.
just some air, maybe. How many do you have in here? 5?
5. I had one person that was emailing me that was trying to get in to be a 6th.
Oh.
But I just a lot of
Did you
them.
guys have any freshmen?
No, not in exercise phys.
Okay. For what?
Just exercise science, physical education.
Oh, yeah.
I have a lot of freshmen.
So did they show up yesterday? Or, no, Sunday?
For other classes, planning.
Or Friday?
I don't
You don't
know.
know.
I don't know. I was in Florida still. Note to self: don't take vacation right before